Hi and welcome to today's lecture about obesity. We are going to talk about the obesity lecture and we're going to try to explain the guidelines related to it. So I made this lecture based on the guidelines. You could go watch them in the studies and then after reading them, take them as a note and then look at my lectures because everything will make more sense too. So first of all, what is the characteristic of white adipose tissue and brown adipose tissue? And what's the differences between them? So the white adipose tissue has one big lipid droplet and is usually found in under the skin, in abdominal cavity, and also in blood vessels. And these are also bigger than the brown adipose tissue, usually ranging from 30 to 70 micrometer. And also, brown adipose tissue has several lipid droplets. So as we said, in the white adipose tissue you have one big lipid droplet, in brown adipose tissue you have several lipid droplets. And also, these brown adipose tissue are rich in mitochondria and have also more capillaries that are supplying them. But why do we call them brown adipose tissue? And it is because they are composed of a high amount of levels of cytochrome C. And this cytochrome C, of course, contains heme groups and iron. And these heme groups and iron is the components that give it a brown color. So, now, more about the function of the white and brown adipose tissue. So the white adipose tissue function is usually carrying the oxidative phosphorylation. So we know that in glycolysis, when we eat glucose and the cell uptake them, we produce glycolysis, and the adipose tissues are the one that carries this function or also have this function as part of them. And oxidative phosphorylation means that it will end at the ETC and provide ATP for us. But it's also active for fatty acid synthesis, although please remember that the liver is actually, I wrote it as the best friend for this metabolic pathway, because fatty acid synthesis happens in the liver mostly, not only in the white adipose tissue, but the white adipose tissue have this also as a function, but it's not the main one. So in the exam, if they ask what tissue produce fatty acid synthesis, don't write white adipose tissue, because it is partially true, but it's not the main tissue. So the main tissue is the liver, of course, we know from Los Colloquium. Also, the fatty acid that we have, the free fatty acid, is used to make DAG synthesis. And the most important part of actually the white adipose tissue is the lipolysis. So when epinephrine or glucagon would bind to its receptor, producing ATP and then CAMP and then activating pKa, and then this reaction, you see it here. We will get free fatty acids and glycerol, which could then be used for glyceroneogenesis or then binding to albumin and then used in beta oxidation or citric acid cycle and producing energy in our body. But also, if we just not consider, so the first function, you could say it's the metabolic function, but the second function is that it's actually also active for, if we can see, it's also considered as an endocrine organ because it stimulates some hormones, and this hormone is called adipokinase, which the main function is to regulate metabolism. So this adipokinase have two hormones related to them. The first hormone is called leptin, and this leptin is secreted by adipose tissue to tell the hypothalamus to stop making synthesis of different metabolites. And actually, in other words, you could say it's just a hormone that tells the hypothalamus that stop eating. We are full, we don't want to eat more, don't consume more food. And we will look through, I think, more about the leptin in details later on. And now the brown adipose tissue, these are located actually near the vital organ that would need heat and energy and warm temperature, because these brown adipose tissue do a lot of beta oxidation. And if these are exposed to cold temperature, these could then be converted to beige adipose tissue actually. But what is the most important, I don't know how most importantly, okay, Don't mind the writing, it's autocorrected. The most important function of the brown adipose tissue is to produce this UCP1. And what is that UCP1? So UCP1 are actually an uncoupling protein. So it uncoupled the ATC. So what happens here is basically that the hydrogen that usually leak out in the ATC and then go back to the ATP synthase is actually not going back to the synthase and is actually going back through this uncoupling protein UCP1. And this will not give us energy, this will not give us ATP, but would provide heat. And this is important for fetus and also babies because they don't have a shivering mechanism. So this is actually the way of them to keep heat in their body. So that's why the brown adipose tissue is very important. So basically it produces this thermogenin. Okay. So, and yeah, as I said, the other function of the white adipose tissue is that these are actually considered as an endocrine organ. I actually like this picture because if we look at leptin, we see here this mice which does not produce leptin is like getting obese. So we could understand from this hormone that leptin actually keep the normal body weight in our body, and if we lack them we will get obese if we don't have leptin production. And the response of leptin binding to the hypothalamus is basically that it will produce norepinephrine and activate protein kinase A, and then tag metabolism and beta oxidation will happen in the mitochondria and we will generate heat. But also, what is interesting is that leptin is actually also affecting another hormones. So mainly neuropeptide Y and alpha melanocyte. So what does neuropeptide Y do? So neuropeptide Y actually would tell the hypothalamus that we should eat more and metabolize less, and this is if we need energy or are hungry. And the alpha melanocyte would do the opposite, tell us to eat less and metabolize more. And this is interesting because leptin have effect on these two, like if you could tell them hormones, because leptin would stimulate alpha melanocyte to tell us to eat less and metabolize more, but would inhibit neuropeptide Y. Also, two other hormones that would affect neuropeptide Y and alpha melanocyte is insulin, telling the neuropeptide Y to eat more and metabolize less. It will actually inhibit the neuropeptide Y, sorry. It will inhibit the neuropeptide Y. And also the PYY and GLP, glucagon-like peptide 1 hormone would also affect the neuropeptide Y and inhibit it. Where we could see the ghrelin hormone, which is a hunger signal, would tell the neuropeptide Y to stimulate it, so we eat more, so we feel more hungry, we want to eat more. You think about you want to eat a pizza or a hamburger. But leptin and insulin and glucagon like peptide 1 and PYY would actually do the opposite. So we would not feel that we want to eat, we just want to sleep, and we basically feel like we have food coma. And this is the interesting part of the leptin hormone. Now, what we also mentioned, the first part of adipokinase is leptin. And the second part, or the second hormone of adipokinase, is adiponectin. And this is a very important hormone. Why? Because it increases sensitivity of insulin for organs, so it helps tissue respond better to insulin. And its main function, shortly, is to burn fat during like energy states, or when we need to burn fat to produce energy. And which metabolic pathway would be affected by this adiponectin? First, in the muscle, it would provide beta oxidation and glycolysis to provide ATP, so to produce energy. But in liver, it would inhibit fatty acid synthesis, cholesterol synthesis, and gluconeogenesis. So we would see lower production of glucose level and also inhibiting of fatty acid synthesis and cholesterol synthesis. And in the liver, it will also promote beta oxidation. So it will promote glucose uptake and lower it, and by using glycolysis and inhibiting gluconeogenesis, because we know that gluconeogenesis produces glucose. But also, and how would this mechanism work? So what would stimulate this adiponectin? What would tell the adiponectin to be secreted and do its effect on beta oxidation, glycolysis, cholesterol synthesis, inhibition, fatty acid synthesis? Basically, it is done via the AMPK pathway. So this is active when ATP is low. So AMPK is like a signaling substance or a molecule. So it signals to the cell to use the ATP which it have stored. And if we want to look at it in details, it actually inhibits the acetyl coenzyme A carboxylase, and this enzyme make malonyl coenzyme A. And inhibiting of this acetyl coenzyme A carboxylate would inhibit malonyl coenzyme A and not let fatty acid enter the mitochondria. And this is in the usual state, but if we inhibit it, then fatty acid would enter the mitochondria, and we know that fatty acid in mitochondria would lead to beta oxidation. So that's why it is a very important signaling molecule, this adiponectin, because it will provide beta oxidation. And this is actually very good for people who are struggling with obesity and type 2 diabetes and insulin resistant. Okay. And what is the causes of obesity? I will not put so much energy for this part, because I think this is even if I take an idiot and explain it to him, I think he will know what would cause obesity. So basically, obesity is when you are overweight, and of course, what would provide you being overweight? An imbalance between food intake and expenditure. So this happened when you would eat very much and you would not exercise, or you not use any energy. Everything is stored in your body. Also, one of the system that would be activated due to overstimulation or over eating would actually increase this mTORC1 factor, and this mTORC1 would do many functions, but it will lead to lipid synthesis, fatty acid synthesis, and so this will lead to storage of many metabolites instead of using them to produce energy, and this is why it's not so good to stimulate this system. Okay, so let's see. We have more to talk about. Okay, so the type of diabetes, I don't think this is so. I think many people know the type of diabetes. So you have, first of all, if no insulin is produced due to the autoimmune attack on the beta cell, this is of course type 1 diabetes. So you don't produce any insulin at all, or a very little. And the type 2 diabetes is that you actually have insulin production, and actually at a very high rate, because the tissues are not responding to insulin. So if you eat a donut, this donut will be broken down by enzymes in the small intestine to monomeric glucose. This glucose will be uptaken by your GLUT2 receptor, and then will be then produced, and it will be used in the cell to produce energy through glycolysis. And this is the normal case. But if you're having a problem with type 2 diabetes, it would mean that when insulin is secreted, this glucose will not be uptaken by the cells, and this would lead then to a very high levels of glucose levels in the blood, which means severe hyperglycemia. Also, you have GDM, which is basically when you have hyperglycemia during pregnancy. Okay, let's see what we have more to talk about. So how is it regulated, and what is the stages of telling that we are having type 1 and type 2 diabetes. So, the first mechanism for both actually the types of diabetes is that our body tries to get rid of it through the urination and we become hydrated and thirsty. And this is like the first signal to everything. And the second signal, sorry, one second. I have a machine that started to work for no reason. The second mechanism of, or the second signs for each type of diabetes is in type 1 we are observing high levels of ketone body in the blood. So acetoacetate and beta hydroxybutyrate. I am very hating on the autocorrect that are not correcting the things that I write, but yeah, let's just pretend that we're not seeing the errors. And this would lead then to elevated hydrogen ions in the blood and decreasing pH, and this is one of the signs of ketoacidosis. So when we see ketoacidosis in the blood, then we know that this is one of the type 1 diabetes signs. And type 2 diabetes is basically when we see very high level of insulin in our body. So usually, like if we say you eat, and this is normal cases right now, if you eat like a donut or sugar related food, then you know for sure that you need insulin for the tissue to respond and uptake this glucose. But if you're a type 2 diabetic, then this would lead to that actually your body is producing too much insulin for just a normal amount of sugar. So you're not eating a high amount of sugars, but your body are like abusing the secretion of insulin. So you have very high insulin levels in the blood. And what would this do? It will actually then of course lead to fatigue of these beta cells in the pancreas. And in the end, no insulin will be produced. And this will lead then to severe hyperglycemia. So this is how you would distinguish between the type 1 and type 2 diabetes. And also other symptoms which we see in type 2 diabetes is high TAG levels and LDL, and high blood pressure, and very high levels of fibrinogen and C-reactive protein for inflammation, but also low HDL levels in our body. And this is how you would also say that this is actually a type 2 diabetes symptom or type 2 diabetes patient, by understanding these symptoms. So the lipid burden hypothesis, basically, so this will be explaining like the overall picture for insulin resistant people, so type 2 diabetic. Basically, if you would overload the adipocyte, you will become insulin resistant. And how would this happen? And this would start with that you eat very high levels of triacylglycerides in the diet, and these are stored in the adipocytes. But the more you store TAG in the adipocyte, the more you store these adipocytes, this TAG in the adipocyte, it will lead that the adipocyte will get enlarged. And what would this do? It will produce a monocyte chemotaxis protein, MCP1, this one right here. And this is very important because what does this do? This does tell the macrophage to infiltrate adipose tissue, so it will kill this adipose tissue. And this is one of the signs of chronic inflammation, as it's written here. And what would this 40% breakage of adipose tissue mass do? So this TNFA would favors export of fatty acids. So when we have fatty acid, this molecule right here will tell these fatty acids to then enter the liver and enter the muscles. And when this fatty acid is entering this muscle and liver, what will happen here is that it will interfere with the GLUT4 movement. And when it interferes with the GLUT4 movement, it will not allow the glucose to be uptaken by the muscle and the liver. And this is why you are becoming insulin resistant if you are obese, or if you are like a very high level of overweightness. And so basically, like, why do we say, like, if you just think about it, if you were an overweight patient, it doesn't mean that you should be a type 2 diabetic. But in these cases, if you enlarge the adipocyte, this is what would happen with you. Okay, let's see what we have more. Now, also, what is the HbA1c in relationship to diabetes, and what are the complications of this should not be located, this should be elevated glucose levels in the blood. So basically, it's when glucose would bind to hemoglobin. So basically, we have glycosylated RBC, and this glycosylated RBC just actually means that you have a hemoglobin with glucose binded to it. So this red blood cell can't transport oxygen to the extremities, and this would lead then to many complications, which is written here. So you have renal damage, cardiovascular diseases, also damage to the nerves and blood vessels, and also would cause retinopathy. Like you could see here every complication. I don't need to be reading that out to you. Just waste of your time and waste of my time. Now, the favorite part of us that study biochemistry is that we love to understand the treatments for the type 2 diabetes, or any type of disease. We are always interested to look at the treatment. So basically, we have one of the, I know I wrote Ozempic, I know it's not written here, but Ozempic is actually one of the type of GLP-1. If you don't want to use Ozempic, use Byetta as an example, or this sitagliptin. It doesn't matter. The thing is that I took a glucagon-like peptide 1 drug. And what does this glucagon-like peptide 1 drug does? It basically enhances insulin secretion by the pancreas, so it will increase glucose uptake, but it would also inhibit glucagon uptake. So what would this do? Or not glucagon uptake, but it will inhibit glucagon secretion. And what would this do? This will lower the blood glucose levels in the body. Glucose level. So this will lower the glucose levels in the body, and also will help increasing insulin. Also, one of the treatment is bariatric surgery, I think it's called, and also training. Very important. Train, because what would this do? It would actually lead to an increased level of irisin hormone, and this irisin hormone would convert this white adipose tissue to actually brown adipose tissue, and then it would provide better oxidation in our body. And I talked about the function of brown adipose tissue earlier on. This is everything that I wanted to talk about today. I hope this lecture about obesity made obesity more clear for you guys what it is. And also we talked about the hormones, and I talked about types of diabetes and the relation for them, and basically this lipid burdens hypothesis also, which is known, and the relationship of HbA1c and relationship to diabetes. Also, what would be the effect of elevated glucose levels, and also lastly how we treat different types of diabetes and diseases. And thank you guys for listening, and if you have any question, just drop a comment or write to me, and I will try to answer your questions. Or if I said anything wrong, also write it in the comments so I could then fix it. because I do these lectures as I also learn, so that's why not everything I said is 1,000,000% correct, but everything should be at least making sense and at least partially correct with what we are learning. And yeah, thank you guys for listening and have a nice day.